by bit, I'm going to talk about the embryology of the meninges. Meninges are not just a covering of the brain. They are important in the formation of the skull, the development of the brain, and have immunological properties. As we all know, there are three layers of meninges in the brain. There's the dura, below the periost. Then there's an arachnoid layer consisting of a very cellular layer lying directly under the dura, and trabecula connecting it to the delicate pia surrounding the brain. We talked about these three layers when we discussed meningeal enhancement. And in pachymeningeal enhancement, there's enhancement of the dura and the outer part of the arachnoid. And in leptomeningeal enhancement, there's enhancement of the arachnoid inner part and the pia. I also mentioned the primitive meninx when I talked about arachnoid cysts in the differential diagnosis of plague spout cyst. And the primitive meninx is established very early in embryology. It divides into an outer and into an inner layer, and eventually gives rise to the three layers of meninges as we know them. The inner layer is called the endomeninx, and the outer layer is called the ectomeninx, and you can recognize them in this 7-week-old human embryo. This is a section through the forebrain with the lateral ventricles and the hippocampal primordium here, so we're anterior of the foramen of Monro. And the exact origin of the primitive meninx has been some debate for a while, and it was unclear whether they were derived from neural crest cells or from mesoderm. And that is because neural crest cells and mesoderm mesenchymal cells are cytologically indistinguishable, and that is because these cells arise in the same region just a few days apart. So the cells are not twins, but they are siblings. So the mesoderm is formed during gastrulation, when there are cells from the epiblast migrating through the primitive streak, transforming when going through the primitive streak from epithelial cells into mesenchymal cells. And a few days later, there's neurulation, there's the neural plate, and the borders of the neural plate become the neural crest. So this happens in the same region a few days apart. And when the neural tube is closed, the cells of the neural crest are going to give rise to neurons of the peripheral nervous system, and also give rise to several skin cells, such as melanocytes and Merkel cells. It has been shown a little bit more than 10 years ago that the meninges covering the forebrain, so the cerebral hemispheres, are derived from the neural crest, and that the meninges covering the brainstem are derived from mesoderm. And this is a more recent article from 2021, where you can also see the parts derived from neural crest and mesoderm in purple. And in the spine it becomes a little bit more complex, and we're gonna look into this in more detail when we're going to discuss meningiomas in a few videos. The meninges do not only cover the brain, but also cover the blood vessels penetrating the brain. And I already talked about that as well in the video on cerebral amyloid angiopathy and the interaction between the basal lamina of the vessels and the basal lamina of the brain. So surrounding the vessels, there's also a layer of pia, and this is the perivascular space important in the lymphatic system, which we will also discuss in the future. And in the past minutes, I've mentioned mesoderm several times. And just to bring back into mind, in the body, the mesoderm gives rise to the bones, the cartilage, and the cardiovascular system. So to all the blood cells. So also the white blood cells, so immunological properties. The same group that described the lymphatic system described a newly fourth meningeal layer in 2023, the subarachnoid lymphatic-like membrane located in the arachnoid reticular space. And they said it was never described before because it disintegrates. And this year there was a very nice article where they retrospectively looked at previous studies, and this was a study from 2021 with a very nice electron microscopic picture of the cellular layer of the arachnoid membrane. The brain, this is the trabecular part of the arachnoid space, and there was this membrane in red that in 2021 was described by Sabori et al. As Paya, but with the knowledge of this mesothelium-like membrane in the subarachnoid space, turned out to be this newly discovered fourth layer. So it has been seen before, but it was overlooked or not noticed or labeled wrong. And the Paya is against the brain in this image. And in the next video, we're going to focus on this subarachnoid space and the trabecula and the formation of arachnoid cysts. So I do hope you will stay tuned.